Hello everybody, we're going to be talking about genes, alleles, and our friends, the little Punnett Squares. So let's do a little vocab, just set everybody up on this one. Gene is any set of instructions that allow you to do something in your body, or for plants to have something going on. Any living organism that does anything, there has to be a set of instructions. Those instructions are called genes. Now, since everybody comes from two parents, you have two variations of that gene. Maybe mom gave you one, maybe dad gave you the other. Those variations of the genes are called alleles. So for instance, flower colors. All flowers have color. That's the gene. The allele part of it is, am I going to be a white flower, purple flower, red flower, pink flower? That's the variation. So how do we figure this out? If we're a botanist and we want to figure out certain genes of these two flowers we have, we want to cross them and make this beautiful color. What type of colors are we going to get out? That's where Punnett Squares come in, and that's what we're going to be talking about right now, how to figure out Punnett Squares. So here's an example Punnett Square. It's talking about white flowers and purple flowers crossing. Now, there's two different things we look at in a Punnett Square. One, it's called the genotype. That is essentially the letters. If we're talking about genotype, we're only talking about the genetic trait. What genes are they getting? What genes are they giving? Capital and lowercase. Remember, capital letters are dominant. Anytime there's a dominant capital letter, that trait is going to be the one shown. The only way you can get a recessive trait like white flowers to appear is if both alleles, aka little p and little p, are both there, and that's the only way you express white. If you have a P, a capital P, it's always going to be dominant, and that's going to be purple. Now, sometimes geneticists, like a botanist, won't want to be talking about, oh, I have two capital P's or a capital P lowercase. They want to be like, I have a white flower, I have a purple flower. That's phenotype. Phenotype is the physical appearance of an organism. What does it look like? I always remember pH, phenotype, pH, physical characteristic. What does it physically look like? Both start with pH. In genotype, well, genes are just letters. So let's do an example of how this whole thing gets crossed. So you can stop it and record this Punnett Square. All right, welcome back. So let's say we're going to throw capital B equals non-bald. So you're not bald. But let's say little b means that you are going to be bald. In this example, we had two individuals have a child. Across the top, the genotype or the letters are capital B, lower b. And once again on the side, it is also heterozygous, which means it's different. There's the little and big, one recessive, one dominant. So both are capital B, little b, both parents are. So what's going to happen? What's the probability of that offspring having male pattern baldness or not being bald at all and having no possibility of being bald? Now, this is where we take our letters and we bring them down. So this letter B comes into this box, and it also comes into this box. Little b comes here and also here. So for this case, we have capital B, little b, capital B, little b. That's coming from the mom. Now, going across on the side, let's bring those letters across here. So that B goes into here. That capital B also goes over here. Little b goes in here. And a little b goes all the way over here. And yes, I'll make this a little bit cleaner. So, so these get interchanged here. So now, I'm going to erase this so we can see it a little bit better. But that's how we get our certain b's in there. So we have to add, oops, I don't know how that got over there, a little b here, because that came from the side. We're going to have to add a big V here. Now, since dominant letters go first, it doesn't matter where dominant letters or capital letters come from, they always go in front. So now here's our Punnett square completed. So we have capital V, lowercase and an uppercase and lowercase, and two lowercase. So our genotype, if we're doing a ratio for this, it would be 1 BB colon, like in math, is a ratio, 2 oops, heterozygote, or capital and lowercase, and finally there was only one little b, little b. So that's our genotype. So that's our ratios, how many of our genes are being expressed. Now, this happens every single time there is a crossing. These, if let's say the capital B and capital B, that's what the offspring gets, it's not like it goes away. Every single time a new offspring has come, these four options are always there. So that's genotype. What about phenotype? What's the ratio for that? So first we'll go with the capital B, which is non-bald. Anytime there's a capital B in here, it's going to be non-bald. So take a second and see if you guys can figure out how many bald versus non-bald there are. All right, we're back. So let's see what you guys have. So for being bald, non-bald, capital B, so non-bald here, I have, looking through our Punnett square, I have 1, 2, 3 that have capital, which means 3 non-bald. And obviously just mathematically here, 1 possibility of being bald. So it's a 3-to-1 ratio. If you want to go and put it into percentages, it's all by 25s. So it'd be 75% probability of being non-bald to 25% being bald for this situation. And for the top, it would be 25%. Oops. I'll move that up in a minute. 25% capital B, 50% little b, capital B, little b, and finally 25% possibility of having both recessive genes. So that is one example of a Punnett square using male pattern baldness in humans, but you can do this with anything that has a trait. And this is a monozygotic cross, meaning we only did one trait, which is baldness. I hope this was informative. If you have any questions, please email or comment, and I'll get right back to you. Thank you, and have an amazing day.